Welcome to our module on erythropoiesis. Erythropoiesis is simply defined as the process of formation of a red blood cell. It is one of the classifications of hematopoiesis, and in particular, it focuses on the production of erythrocytes, hence the name. We'll talk about the different aspects of erythropoiesis, from the stages to the processes to the duration, outcome, regulation, and some pathologies. The entire process of erythropoiesis begins with the stem cell stage. The stem cell is known as the hematopoietic stem cell. This undergoes cell division and differentiation to form progenitor cells. These are the two progenitor cells in erythropoiesis. They include the boss forming unit erythroid and the colony forming unit erythroid. These two undergo differentiation to become the precursor cells, and these are all the precursor cells under erythropoiesis. They include the pro-erythrocytes, the basophilic erythrocytes, polychromatic erythrocytes, and orthochromatic erythrocytes. And once each of these forms one another, they would eventually give rise to the formation of reticulocytes. Reticulocytes are a form of mature red blood cells with presence of nuclear materials. Erythrocytes would eventually undergo maturation to form the mature red cells, or what we call erythrocytes. Let's talk about the process in depth, starting with the stem cell stage. Red blood cells are usually derived from a form of stem cell called pluripotent hematopoietic stem cell. These are stem cells that typically can give rise to any organ or structure tissue in the body. A pluripotential stem cells tend to have two major properties. They have the ability to undergo self-renewal, and they have the ability to undergo cell differentiation. And when they self-differentiate, they could either self-differentiate into a myeloid, or what we call a germ committed stem cell, or a lymphoid committed stem cell. The germ committed stem cell means granulocyte erythroid macrophage megakaryocyte committed stem cell. That's the full meaning of germ, GEMM. So when focusing on erythropoiesis, it's important to note that the pluripotent hematopoietic stem cells tend to differentiate into the myeloid or the GM committed stem cell. And this GM or myeloid committed stem cells would eventually differentiate into either of these three progenitor cells. So they could either differentiate into the boss forming unit erythroid, or the boss forming unit megakaryocyte, or the highly proliferative potential colony forming cells. Each of these cells are progenitor cells for different lineages. So the boss forming unit erythroid is the progenitor cell for erythropoiesis, and that's what we'll focus on in the next slide. The boss forming unit megakaryocyte is the progenitor cell for megakaryocytopoiesis, and then finally the highly proliferative potential colony forming cells is the progenitor cell for the process of granulopoiesis and monocytopoiesis. In terms of erythropoiesis, our focus will be on the formation of boss forming unit erythroid. We'll talk about that in the next slide. So boss forming unit erythroid is the first progenitor cell of the process of erythropoiesis. How we tend to recognize it, although it is morphologically is not morphologically identifiable, and it can't self-renew. How we tend to identify it is by the presence of some receptors on its membrane. Usually, the boss forming unit erythroid is already committed to the formation of erythrocytes. And how we tend to know that is that it expresses erythropoietin receptors on its membrane. However, it lacks hemoglobin, and it contains a large amount of RNA for protein synthesis. This boss forming unit erythroid would undergo cell differentiation and eventually give rise to a new form of cell called colony forming unit erythroid, which we'll talk about that next. The colony forming unit erythroid is the second progenitor cell in the process of erythropoiesis. Contrast to the boss forming unit erythroid, it expresses more erythropoietin receptors, and that's what we use in distinguishing it from the boss forming unit erythroid. It also lacks hemoglobin. It also contains RNA for protein synthesis, and similar to the boss forming unit erythroid, because it is a progenitor cell, it is not morphologically identifiable. It cannot self-renew, and it would automatically undergo cell differentiation to give rise to the first precursor cell, which is the pro-erythroblast. Proerythroblast is the first precursor cell of erythropoiesis. It is a large cell, usually the largest of the precursor cells, that measures about 24 to about 26 micron size. It appears a dark bluish under the microscope. I'll tell you the reason for this. It tends to contain a large nucleus, which is described as a coin-shaped nucleus. It has a perfect coin circularity as outlined in this image, and this form or shape of the nucleus tends to follow the rest of the different precursor cells. So all the other precursor cells tend to have a similar shaped nucleus. A large coin-shaped nucleus is seen in all of those precursor cells. The cytoplasm of the proerythroblast tends to be rich in RNA, and please do note that the presence of RNA or hemoglobin in a cell tends to make it either stain bluish or stain pinkish, and I'll talk about that very soon. So the proerythroblast tend to have a lot of RNA, whereas have very little hemoglobin. In fact, hemoglobin production usually begins in the proerythroblast stage. So in terms of the cytoplasm and the stain, why proerythroblast tend to stain bluish, dark bluish, is because normally when we are staining cells and viewing them under the microscope, we either use an acidic stain or a basic stain. Whenever a cell has a cytoplasm rich in acidic properties, usually acidic properties would be properties of RNA or DNA. So whenever there's a lot of RNA in the cytoplasm, that cell has acidic properties in the cytoplasm. A cell like that tends to stain with basic stains. So basic stains tend to attract acidic structures. So and that will make the stain to be appearing bluish under the microscope. So for example, proerythroblast tends to have largely RNA material, so it's largely acidic. Because it's largely acidic, when we stain it, it tends to attract basic dye or basic stain to it, making it to be bluish in appearance under the microscope. That is in contrast to cells that tend to have largely basic structures within their cytoplasm. So hemoglobin, for example, is a protein. Protein contains largely basic structures, and because it contains large basic structures, whenever a cell contains a lot of basic structures within it, that cell tends to attract a lot of acidic stain to it, and they tend to appear um, reddish or pinkish in color under the microscope. So please keep that in mind, because as we go ahead to talk about other precursor cells, you see that there's a change in the cytoplasm content, and eventually there's a change in the appearance under the microscope. The proerythroblast tend to automatically divide, undergo cell division and cell differentiation to give rise to the next precursor cell called the basophilic erythroblast. We'll talk about that in a minute. The second precursor cell is the basophilic erythroblast. It is smaller in size compared to the proerythroblast. It also appears bluish under the microscope, but it is less bluish compared to proerythroblast. And this is because of the content of the cytoplasm. The cell of the nucleus of the cell remains large and remains perfectly shaped as a coin circularity. However, the cytoplasm of a basophilic erythroblast is rich in RNA and has more hemoglobin compared to the pro-erythroblast, and this is the reason why it stains less bluish compared to the pro-erythroblast. Usually, the basophilic erythroblast has two stages or two substages. This includes the basophilic erythroblast type 1 and basophilic erythroblast type 2. The difference between both of them is that the basophilic erythroblast type 1 is larger in size, is less deeply bluish under the microscope, and has fewer hemoglobin, whereas basophilic type 2 erythroblast tend to be smaller in size. It is more deeply bluish and has more hemoglobin compared to type 1. Basophilic erythroblast, as you can see under the microscope in this image, you can see it's bluish but not as bluish as compared to the pro-erythroblast. Basophilic erythroblast undergoes division and differentiation to form the third class of precursor cells under erythropoiesis, known as polychromatic erythroblast. We'll talk about that in a minute. So the polychromatic erythroblast is the third precursor cell under erythropoiesis. It is smaller in size to the basophilic erythroblast, which we talked about in the previous slide. It appears multicolored under the microscope, and the reason for this is because of the presence of large amount of RNA and hemoglobin in the cytoplasm. So hemoglobin and RNA are usually in almost equal amounts within the cytoplasm. And as a result, because both of them are basic and acidic, they tend to attract both basic and acidic stains respectively, leading to the appearance of a mixed color, the mixed reddish bluish color under the microscope. Polychromatic erythroblasts tend to undergo cell division and differentiation to form the fourth cell under the precursor cell stage of erythropoiesis, known as autochromatic erythroblast, and we'll talk about that in a minute. Autochromatic erythroblast is the fourth precursor cell in the stage of erythropoiesis, and unlike the polychromatic, it is smaller in size. It appears reddish or pinkish under the microscope, and this is by virtue of what is found in cytoplasm. On the cytoplasm of the autochromatic erythroblast, there is large amount of hemoglobin and very small amount of RNA. So this is a reversal of what we are seeing for basophilic and polychromatic. And the large amount of hemoglobin, because hemoglobin is a basic structure to stain with acidic stain and give that reddish or pinkish picture appearance under the microscope. The reason why we tend to have less amount of RNA is because of what happens to the nucleus in the autochromatic erythroblast. So autochromatic erythroblast usually begins with that coin-shaped nucleus that we talked about in the other two stages. But with time, this coin-shaped nucleus undergoes a particular process called pycnosis. So pycnosis is the eccentric removal or displacement of the nucleus of the autochromatic erythroblast, in which it comes out or it's ejected out of the cytoplasm of the cell, and eventually this makes the autochromatic erythroblast to be without a nucleus or to be anucleotic. For this reason, we usually see the autochromatic erythroblast is incapable of undergoing further mitotic division, because for mitotic division to occur, for mitosis to occur, we need the nucleus of the cell to undergo cell division, to undergo DNA replication and automatically cell division. So the process of pycnosis of the nucleus of the autochromatic erythroblast makes it or renders it to be incapable of further mitotic division. And whenever we have that pycnosis occurring, where the nucleus is being removed from the cytoplasm of autochromatic erythroblast, we say that the autochromatic erythroblast has matured into a reticulocyte. We'll talk about the reticulocyte in a minute. So reticulocytes are the mature form of the red blood cell lineage, but they are different from a mature red cell based on a few things. First of all, the reticulocytes could either be found in the bloodstream or found within the bone marrow. They're largely found in bloodstreams. They migrate from the bloodstream into the spleen to undergo final maturation. And what does that entail? So usually reticulocytes tend to contain fragments of nucleus or nuclear materials within the cytoplasm. Remember that for autochromatic erythroblast to undergo maturation, pycnosis must occur. Pycnosis that tend to occur in autochromatic erythroblast tends to be incomplete. There are very few nuclear materials found in the cytoplasm of the reticulocyte after pycnosis occurs, and those very few nuclear materials will need to be thoroughly removed after the reticulocyte comes out of the bone marrow. So reticulocyte enters into the spleen, and it takes about 48 to 72 hours for it to undergo full maturation. Maturation in the spleen just involves the removal of those fragments of nuclear materials that are left over within the reticulocyte once it has made its way into the bloodstream. And at the end of this stage, at the end of the removal in the spleen, the reticulocyte will be converted into a mature erythrocyte, or what we call a mature red cell. The entire process of erythropoiesis from the pro-erythroblast stage to the mature erythrocyte stage takes about 7 days, and from the reticulocyte stage to the mature erythrocyte stage, this takes about 2 to 3 days within the spleen. This is just a picture showing us erythropoiesis from the point of the precursor cell to the mature red cell. So it takes about 7 days for a pro-erythroblast to give rise to a red cell, and we usually take note of the fact that one pro-erythroblast usually undergoes about 4 mitotic divisions to form 16 reticulocytes. So this is very key to note. I'll tell you how we arrive at this figure in the next slide in terms of the division that the pro-erythroblast and its fellow precursor cells tend to undergo. So we go from one pro-erythroblast undergoing division and differentiation to form two basophilic erythroblasts. Then the two basophilic erythroblasts will give rise to four polychromatic erythroblasts once it undergoes cell division and differentiation. Then the four polychromatic erythroblasts undergo cell division and differentiation to form 16 autochromatic erythroblasts. Remember that the autochromatic erythroblast does not undergo further mitotic division because of pignosis. So four we have the 16 autochromatic erythroblasts undergoing maturation to form 16 reticulocytes. Then the 16 reticulocyte comes out of the bone marrow into the bloodstream, enters into the spleen, undergoes full maturation to form 16 erythrocytes. So one pro erythroblast gives rise to 16 mature red cell, 16 mature erythrocytes. And this is practically how those divisions tend to come to play. So we have four mitotic divisions occurring between pro-erythroblast, basophilic erythroblast, polychromatic erythroblast, and the autochromatic erythroblast. The entire process of erythropoiesis is regulated by a number of factors. This includes erythropoietin, which is the most important factor being produced from the liver and the kidneys. Kidneys produce about 85% of erythropoietin. Same with the liver, producing about 15%, and erythropoietin tends to drive the production of red cells through erythropoiesis. Other factors that are involved in erythropoiesis include erythrocyte growth factor. These are factors found within the bone marrow matrix of the heme space. We also have the granulocyte macrophage colony stimulating factor, stem cell factors, and interleukin 3. These are all factors involved in regulating erythropoiesis. What happens whenever we have erythropoiesis being abnormal in terms of its process? So you can have four major things occurring. We could have an increased production of red cells during erythropoiesis. This could lead to a condition we call polycythemia. We could have a decreased production of red cells. This leads to a condition we call anemia. And in terms of the finer details of the process, we could have abnormalities in DNA synthesis, replication, and cell division. This could affect production of red cells or the process of erythropoiesis and lead to the formation of what we call macrocytic or megaloblastic anemia. Or we could have problems with the formation of hemoglobin, which will lead to the production of what we call a microcytic anemia. So how does the last two tend to come about? So please do note that when we go from the progenitor cells down to about the first two precursor cells, so from the progenitor cells, the boss forming unit erythroid, the colony forming unit erythroid to the pro-erythroblast and the basophilic erythroblast. Usually, what tends to drive that process is DNA synthesis. So DNA synthesis and cell division drives that process um, all the way down to the autochromatic erythroblast. But from the pro-erythroblast all the way down to autochromatic, another set of processes is is occurring, and that process is the process of hemoglobin formation. So as we go down the chain, we have red cells undergoing cell division, and this cell division is driven by DNA synthesis. Without DNA synthesis, we cannot have cell division occurring. And as we go down, we have the cells becoming smaller in size. This is very important to note. So the pro-erythroblast is larger than the basophilic erythroblast, which is larger than the polychromatic erythroblast, which is larger than the autochromatic, which is larger than the reticulocyte, which is larger than the red blood cell. And that's how division tends to make sure that occurs. Also, as we go down, we have an increase in the amount of hemoglobin. So we have hemoglobinization of the cytoplasm occurring. So the pro-erythroblast has less hemoglobin compared to the basophilic erythroblast, which has less hemoglobin compared to the autochromatic, which has less hemoglobin um, compared to the reticulocyte. So as we go down the process of erythropoiesis, we have more hemoglobin being formed or being present in the cytoplasm, and this is very key to note. So what happens in terms of problems with DNA synthesis and problems with hemoglobin production? We'll talk about that in the next slide. It could lead to different forms of anemia. It could lead to a megaloblastic or macrocytic anemia or a microcytic anemia. Whenever there's a problem with DNA synthesis, replication, or cell division, this leads to macrocytic megaloblastic anemia. So how does this occur? Usually with DNA problems, we have less mitotic divisions occurring. Because there's less mitotic divisions, instead of having about 4 divisions, we have less than 4 divisions. Because we have less than 4 divisions, we have smaller amount of reticulocytes being formed. But more importantly, because we're having less divisions, we have the cells not being smaller in size. So we have the newer cells that are being formed not being smaller in size compared to the previous cells. Remember that pro-erythroblasts tend to be larger compared to basophilic. Basophilic tend to be larger compared to polychromatic. Polychromatic tends to be larger compared to autochromatic, and all these tend to occur when it's 4 divisions or 4 mitotic divisions occurring in the process of erythropoiesis. Whenever there's a problem with the DNA synthesis, replication, or cell division, this affects the number of divisions, and this automatically affects the size of the cells that are being formed because we have larger cells being formed at the end of the day. And these large cells that we refer to them as macrocytic cells. So you can see the picture of a macrocytic cell here. You can see the red cells are being very large compared to others. Although also we also tend to know that these cells are megaloblastic. The reason why we say they are megaloblastic is because of the fact that they still have remnants of nuclear material. Because remember that during the process of DNA synthesis, replication, and division, when we get to the autochromatic stage, that particular process is halted and we have picrosis occurring. Picrosis involves the removal of nuclear material. So if that process of DNA synthesis, replication, and division is affected, maturation of autochromatic cells will be impaired, and we don't have complete removal of those nuclear materials, making the cells to be megaloblastic in the sense that their nucleus becomes very large and hard to remove, and remnants of those nucleus will be found within the red cells. So in the case of hemoglobin production, we tend to have microcytic anemia occurring as a result. And this is because of the fact that because there is a problem with hemoglobin production, the process of protein production will be doubled down by the cell. So process of protein production, remember, involves DNA being converted to RNA, RNA being converted to protein. And if the protein is not being formed, then there is a positive feedback that allows for more DNA replication and transcription, which automatically leads to more cell division occurring. So we have more cell division as a form of compensation for the hemoglobin production that is being impaired. So practically, if there's any condition that leads to hemoglobin production being impaired, we have more mitotic division, usually more than 4. We have more reticulocyte formed, more than 16, and automatically, because there's more cell division, we have smaller cells being formed. Because remember, when we go from pro-erythroblast down to autochromatic erythroblast, we have the cells being smaller. So with more divisions, we have more smaller cells being formed. And these smaller cells are known as microcytic cells. As you can see pictures of microcytic cells in the image below. You can see cells are smaller in size compared to a normal red cell. Usually what tends to lead to abnormal hemoglobin production is a number of things, but the most common is iron deficiency. That concludes our module on erythropoiesis.